This immunohistochemical and histopathologic protocol can be used to examine folate receptor beta, which is a candidate protein with potential prognostic and therapeutic value to the vascular microenvironment in giant cell arteritis. This protocol is time-tested and widely employed and can be used to explore the folate receptor beta attributes in temporal artery tissues even after long-term storage.
To acquire tissue sections, use a microtome to slice 4 to 5 micrometer thick sections of paraffin-embedded temporal arteries, floating each section in a 40 degrees Celsius water bath as they are obtained to remove wrinkles. Use glass microscope slides to capture 3 sections per slide, and warm the slides on a warming plate in a 60 degrees Celsius oven for 60 minutes. When the sections have adhered to the slides, place the slides into a vertical rack for drying at 37 degrees Celsius for 24 hours. The next day, transfer the slides to a slide container for storage at 4 degrees Celsius for at least 12 hours. Next, load slides into a slide rack and hydrate the tissues with 2 5-minute 100% xylene immersions and a descending 5-minute per concentration ethanol immersion series with 10 seconds of agitation every 30 seconds. After the 70% ethanol immersion, rinse the slides 2 times in double distilled water for 5 minutes with 10 seconds of agitation every 30 seconds.